Hello, everyone. My name is Viola Rudert. I am the International Distribution Manager at Unchained Labs, and today I will be your moderator. I'm very glad you're joining us. Before I introduce Rifar, I would like to just tell you that we do have a Q&A session at the end of the presentation. To ask questions, all you need to do is click into the Q&A in the Zoom navigation bar at either the top or bottom of your screen and type in your question. I will, or Rifar will get to as many of them as we have time for. So now I would like to introduce Rifar Georgiou, our so amazing Director for Field Applications in Analytics for Unchained Labs. Today, Rifar will talk about how the Unchained Labs instruments perform for conformational stability measurements, colloidal interaction, self-association propensity with B22, diffusion interaction parameters with KD, just to characterize the effect of biophysical screenings to establish developability and stability. He will also show how these and other Unchained tools can be used for things like buffer exchange or to create different buffer conditions that then can be screened, as well as for in-process and final quality control. Now I'll hand it over to Rifar.
Thank you very much, and welcome to everyone. So my plan is to go over, as Viola said, some of the Unchained tools for the development of antibodies and biosimilars. Before we get started, I think it's important to put things in context. And I think when you think of antibody development, it's really similar to finding the golden egg, right, among many very common eggs. And typically, as you begin the process, you start with a very high number of clones or a very high number of candidates, and through a series of sophisticated processes and characterization, you down select your way to hopefully the most suitable, the most impactful, the most therapeutically valid and potent candidate, which becomes eventually the drug that you commercialize. And so what we're going to be doing here is going over some of those challenges, but also some of the tools that Unchained makes available to you so that you can actually overcome some of those challenges and easily characterize those samples. Okay? When we think about antibody development, there are lots of challenges, right? From very simple ones, which is essentially assessing the concentration, right? So quantifying them, to much more interesting ones where you have to look at the unfolding properties of your samples. Are they still folded? And therefore, are they still therapeutically valid? Do I have an event of aggregation that I have to be mindful of, but also long-term stability? As you think about formulation screening or excipient screening, we're going to be mindful of assessing how exactly our samples perform over time in different conditions, right? And finally, as you make your way down the process, you're going to also need to assess long-term developability, but also scaling and production. And we'll see exactly how Unchained can help you do so. But no matter how you think of this, all of this essentially is defined by time, right? And the faster you can get through those stages, the less you have to repeat them as well, the more efficient your process is going to be, and therefore the more value you are going to derive as you produce your antibodies. So here are three of the initial stages that you can think about when it comes to antibody development. So initially you have essentially a screening that happens where you do some ligand binding, and you're trying to essentially do some functional assays where you're trying to assess how strong of an interaction that you have. You're then going to look for essentially quality, right? How good of a sample do you have as you go through different formulation screens and trying to assess whether or not your protein is still folded and whether you have any aggregation present as you do so, okay? And so the example here show you three world-renowned soccer players or football players, right? And what's interesting about them is that each of them are superstars in the game, but they have very unique skill sets and very unique ways of playing the sport, okay? And so when you think about our technologies, it's pretty much the same. Here are three players, three essential tools that you're going to use to characterize antibodies. And we're going to see exactly how each of them is very unique, but each of them comes together to give you essentially the ensemble of what you need for characterization. The first one is going to be the uncle. And so the uncle is a tool that is used to assess stability, right? And it does so by essentially controlling temperature. And we'll see as well as the other detector is present there, and that's the uncle. In the middle one here, you have the stunner. The stunner to me is about antibody quality control, right? And so as you're producing antibodies, as you are taking them through different stages, at times you're going to quickly want to assess their concentration, but you're also going to assess whether or not they're about the same size and whether you have aggregates present that are beginning to form. And this is how the stunner can be useful in that capacity. It's a small volume, but a very useful way of assessing the quality of your proteins very quickly. And finally, the lunatic, which is going to be high throughput concentration platform. And again, you only need 2 microliters, but we've added something really interesting, which is an unmixed application, which allows you to further look at the impurities and the quality of your samples without having to do additional tests. Yeah. So let's take a closer look at this one. So the OnCall itself is a multiplex system. And when we say multiplex, we mean that there are multiple detectors in one box. What we've done here is taken fluorescence, allowing you to measure the full spectrum from 250 nanometer to 720 nanometer of your sample, and you're able to assess the melting temperature, as well as have a clear idea of how folded or unfolded your protein is in different conditions, okay? The second detector is static light scattering, or SLS, and this one allows you to assess the presence of aggregates, but specifically the temperatures at which aggregates can begin to form and the extent to which you have more aggregates or less aggregates in different formulations, right? And then finally, the third detector is DLS, or dynamic light scattering. And this one is about looking at the exact size of your proteins, your main protein of interest, but also asking whether or not you have aggregates present in that sample, and also being able to determine the relative proportion of aggregates that you have relative to your main protein of interest. Okay? And so all 3 of those detectors exist within the OnCall platform. What makes it unique is that we've added the ability to control temperature. You can also ramp up your sample from 15 to 95 degrees Celsius, or you can use a single temperature and control it precisely within a precision of 0.01 degrees Celsius per minute, for instance. Okay, degrees Celsius. And so when you think about the actual sample itself, right, each sample holder is rather unique. We call the sample holder, which is the black part that you see there, the uni, and it's a cassette or a cartridge of 16 9-microliter quartz cuvettes. And each of those are high quality grades, so they're fantastic for UV or light transfer. And they're also, as you can see, on this black cartridge, which is optimal for heat transfer. And so each of them can be individually filled. You can use 1 uni or 1 cuvette, or you can use all 16 of them, right? And once you've done so, you can then assemble them and seal them into the blue frame that you see there and the silicone seals, which means that once they are sealed, they're completely inert. You won't have to worry about evaporation. You can also do long-term stability studies, meaning that you can hold a constant temperature for extensive amount of time in the instrument, or you can take the sample holder, put it in your oven, or flash freeze it, and then come back periodically to the instrument and then read the actual sample itself and perform a full experiment. So each of them can hold 16 samples. As you can see on the right-hand side, 3 of them can fit in the instruments. So the maximum throughput is 48 samples per run. So very, very good throughput there for a melting temperature, a temperature of aggregation, and dynamic light scattering at the same time platform. Okay. So here is a poster, if you want, of some of the more popular applications that the OnCall platform can support. As of right now, we can support up to 13 applications. The one I have selected here in green is the most popular application. It's the TM, Tagg, and DLS. So what this one consists of is the ability to measure melting temperature, your temperature of aggregation, and a full dynamic light scattering experiment before and after at the final temperature of your sample in one run, essentially one experiment. You don't have to reload your samples. You don't have to repeat it or use different platforms. One platform, you give us 9 microliters, we'll give you 8 data points for each of those samples. As you can see here, there are a number of other applications. So we can do B22KD, which has become quite popular. We can also do isothermal studies, as well as refolding studies, and a number of other applications, as we'll see here. But for the next few slides, I'm going to focus on the TM, Tagg with DLS, and we'll see exactly how that application can be really useful in allowing you to distinguish samples and do some construct screening. Okay. And so before we do so, I want to really give you a bit more insight into what we collect in the TM, Tagg experiment. The raw data for this experiment is a full spectrum, as we stated, from 250 to 720 nanometer. What you typically expect to see is that as your protein unfolds, you're going to have a spectrum here, which is characteristic of the relative unfolding of your protein as the microenvironment around the tryptophan or the aromatic amino acids begin to change. And as they do so, you'll begin to see that the spectrum itself may change in a number of ways, and you don't have to know exactly how it works. The software will take care of it completely. Just know that no matter what happens, because we have a full spectrum, we can characterize just about any conditions, any protein that you may handle in your lab throughout your development, okay? In addition to that, we can use the intensity at 266 and at 473 to give you the temperatures of aggregations, as we talked about. So let's go through an example of protein engineering. This is from our customers at Jensen. And you'll see essentially what they've done here is outline some of the things that they do to down select some of their candidates. And so you can imagine that they start with hundreds of samples at the beginning and they do some binding assays. And as they make their way down through some of the functional evaluations, they can begin to look at some of the thermal and colloidal stabilities of those samples and long-term developability, okay? And so all of these, as you can see, consist of a number of applications, right? Each of those have their benefits and shortcomings. Each of those also have a number of complications and requirements, which also doesn't always make them ideal for every situation. So let's see exactly how the Unchained platform can be useful. What you are viewing here are three antibodies, right? So you have a NIST standard, a NIST antibody that is really well known and characterized. And then you have other wild types antibodies, the so-called mAb1 here and the mAb2 that have been characterized. What you can see is that from that single experiment, right, we're able to not only assess the melting temperatures. In this case, there were two temperatures that we noted here, TM1 and TM2. And then we also were able to measure the temperatures of aggregation at 266 nanometer and at 473 for the samples. I want to bring your attention to one very particular interesting observation here, which is that in the case of the NIST mAb, the melting temperature number 2, the TM2, corresponds very nicely to the temperature of aggregation. And what this tells you is that it is this unfolding transition here which is responsible for the aggregation that you see there. It's a very unique insight that only the Unchained can give you, because we measure both unfolding and aggregation at the same time. Yeah? When it comes to the second and third antibodies that you see there, the so-called mAb1 and mAb2, you'll note something interesting here where the first melting temperature, right, the TM1, is a bit closer to the Tagg, and that's going to be an indication of the effect that this one can have on the aggregation that you see there. And finally, you can see again that the TM2 for the mAb2 corresponds to the aggregation there, giving you a direct indication of what transition or what portion of your antibody, right, of that structure is responsible for the stability of your sample. And so if you are screening different clones or different mutations, you can know exactly which modifications are responsible for your stability, and therefore you can make intelligent choices as you begin to design or engineer your antibodies in early stages. So what we're not showing you here is that those samples were measured by DSC, and the data correlated very nicely with DSC. And of course, we've run DLS on them, and we can see exactly the size and quality of the samples, and they were quite good. The takeaway message here is that if you are doing this by DSC, you will only get one information, which is the melting temperatures, right? And so you'll have no idea what's happening when it comes to the aggregation. You also will not have the ability to screen as many samples. Typically, DSC is a low throughput measurement, and therefore you can only screen maybe 4 to 8 samples if you're very productive in a day. We can screen as many as 144 samples in 8 hours using an average set of settings, if you want. And so on those samples, we can also do what we call B22KD. So B22KD looks at the so-called protein-protein interactions, right, or the diffusion parameters of the samples. It's a very nice way to look at the long-term stability of your samples or the propensity to aggregate. And so what you're viewing here is that typically for a sample, if the KD value is positive, that is good. That means that your samples have a tendency to repulse each other, and therefore they're going to diffuse away from each other and not aggregate. If they have a negative value, that means that they are going to be more prone to aggregation, and therefore you should either stay away from those formulations or those candidates, or you may want to at least engineer them differently. If you look at the example here, we can see significant differences where mAb1 has a negative value of negative 19.5, whereas the CNC0607 has a value of negative 60.7. This is a much worse candidate, right, than the other two. And so if you had to select one, certainly you would select the mAb3 first, number 2 mAb1, and your last one would be the CNC0607, okay? And the B22 data, not shown here, but had the exact same trend, and those give you very unique insight into both the protein-protein interaction for the B22 and the protein-solvent interaction for the KD, allowing you to make very intelligent decisions when it comes to selecting the right mutation or the right clone for your proteins as you select them. Okay. So let's take a step back and think about where does the uncle fit, right? So what I've shown you so far essentially is an example of construct, right, development, so early stage screening. And so if you are looking to assess the right candidates, down select them and figure out which ones have the most suitable properties, the uncle can certainly help with that. In fact, some folks are using it for thermal shift assays to look at even some interactions or some pseudo binding assays. So there are very nice ways in the early stages to use the uncle to assess the quality of the clones that you are selecting, okay, or the constructs. As you make your way downstream to formulation, certainly you're going to be mindful of the effect of excipients, different conditions, different pH, long-term stability, but also freeze-thaw cycles. Imagine being able to load your sample and take the exact same sample that you're going to use in the instrument, freeze it, and then directly put it back in the instrument to assess it and do that over a number of cycles and use the exact same sample as you do that over different time points and over different freeze-thaw cycles. That's what the sample holder and the uncle platform offer you. And finally, as you make your way downstream and you're going to begin to produce it, you want to assess the quality control of your samples. The uncle can do that, and it is in fact 21 CFR Part 11 compliant today. So now let's look at the stunner. So as I mentioned before, the stunner has the ability to measure concentration, right? But it also has DLS built in. And so DLS, or dynamic light scattering, is about thinking about quality. I'm going to measure size. I'm going to assess aggregation in my sample and begin to see whether or not, as I perhaps switch formulations or as I do different things to my sample, whether the actual quality of the sample is changing. The sample volume for every measurement is always 2 microliters, right? So this is completely a small volume or a low volume platform, and you can do high throughput screens, or you can do just one sample at a time. It's extremely fast. So for 96 samples, you can read all of those samples in about anywhere between 5 minutes if you're trying to do just concentration, or if you want to do concentration in DLS, it takes less than an hour to screen 96 samples. So it's really impressive to think about the fact that you can screen samples in less than 30 seconds for each. So what's unique about it is the actual sample holder, or the plate, right? So we have those cuvettes that allow you to read directly there, and they're very, very small, and we can then use that to measure both the concentration and the DLS of the samples at the same time. And so as we stated, you can screen samples very quickly, but you can certainly, if you do DLS, do so in about 1 hour and make it much more effective than anything else that you may encounter on the market. And the stunner is completely automation ready. So if you want to set it up with a system that can do liquid handling or prepare samples for you, you can do so. And we can discuss those offline if you have any questions. So some of you may know what DLS is about, but briefly, I'm just going to remind you, right? So DLS essentially is about looking at size of your particles, but it's called polydispersity. So that second term there, polydispersity, looks at essentially it's a way of thinking about homogeneity, right? So how many of my samples are going to be close to the size I'm expecting, but do I also have a second or third size population or group, right, in my sample? And that's essentially what we are trying to assess. And so if you want to look at the effect of aggregation over time, if you are stressing your samples, or if you are storing them, or if you are doing a lot of buffer exchanges, it's a very useful way of doing so. And so when you think about the different things that can happen to a protein, quite a lot can happen, right? They can be stressed in many ways. From the very early stages, once you've selected your candidates that you're going to move downstream, if the temperature at which they're stored begins to change, that can have an impact on the formation of aggregates. If you're going to buffer exchange them as you're doing your formulation screen, that can also have a very significant impact on the aggregates. right? And of course, as you begin to think about moving downstream to manufacturing and other factors, there are lots of things which can be sources or reasons why aggregates begin to form. And so being able to assess them is going to be very useful. Now, if something changes, what does that mean for you, right? So we have a number of techniques here. You can see the SPR, which is going to look at some of the affinities, if you want, DSC, which is going to look mostly at melting temperatures, right? And then we can do the functional assays. And as we stated, the uncle itself, the very last one, can also offer you some very interesting bit of information on the samples. But as they begin to change, a lot of things can happen and you may not necessarily get the right picture. And that's when the standard can be quite useful in that example or in that capacity. So what I'm showing you here is an antibody, which was stored in a salt formulation at room temperature. And we simply monitored that sample over 3 weeks or 21 days. If you look at that sample by SEC or size exclusion chromatography, you can see that you have an interesting change here, right, in the absorption, if you want, the absorbance of your collections or your fragments as they're coming off your columns, right? If we now look at those samples by DLS, let's see exactly what we see and how much clearer that picture is. On day 0, we can see the actual average size of our samples at about 11.7 nanometer. By day 6, we can see that the size has increased at about 17.2, and the PDI also has increased, which is indicating to us that we are going to have, in fact, a second population here, right, aggregates that are forming. By day 14 or 2 weeks later, we have essentially gotten nearly close to twice, nearly, the size of our samples, and the PDI itself again continues to increase, and we see a slight increase in the second peak there. And after 21 days or 3 weeks, we can see that we have doubled in size. Our PDI also has gone up a little bit, but look at the size of your secondary peak there, indicating that you have a greater proportion of your sample, which is now aggregated, okay? And this is what the standard can do for you. In addition to that, we can look at the UV vis and see that the concentration itself over time begins to change as your protein may undergo other processes and break down or simply hydrolyze, for instance. Okay. We can also see that there are other effects that we can note. So shaking, right? So if you're taking your samples and you're going to move them from one place to the other, are they susceptible to aggregation? Some samples are more likely to aggregate than others. So if you're looking at the example here, we have one sample in a histidine buffer, and we can see the size before at 11.3 nanometer. And after, we can see that after shaking, right, we get a sample that is quite large at about 253.4 nanometer with a PDI that's well above 2.2, which is an indication that you have, in fact, large aggregates present in your sample. As you can see here, things are well over 1,000 nanometer. If you look at the second half of the slide here, you have essentially the same sample, but this time we've added PS80 or polysorbate 80, sorry, the surfactant. And you can see that the size is about the same at the beginning, but even if we shake that sample afterward, we still remain around the same size, indicating that in this case, this surfactant is able to essentially mitigate or reduce the impact of shaking on the formation of aggregates on those samples. If we look at storage conditions again, this is no stress added, simply waiting and seeing the impact over time. You can see again how things are changing, and this is similar to the data set that we saw previously here. Okay. And so what does the standard make sense, right? So it's a tool that measures concentration, but it's also a tool that gives you an indication of the quality of your sample, whether or not you have aggregates. So at early stages, certainly as you are going through lots of candidates, you're going to want to know quickly how much of them you have and to know how much you can use for your analysis. That's very useful. As you make your way down through the workflow of antibody development and you're going to start doing your formulation screening, you're going to be mindful of not just knowing your concentration, but you also want to know whether you have aggregates present and to which extent they're impacting your sample, right? And different method developments, different storage conditions, freeze-thaw cycles, different sources of stress can really impact your samples. And we can see exactly how that can be done or impacted directly using the stunner. And finally, as you make your way to manufacturing, you're going to want to have a QC platform available to you, something that's very quick and easy to use. Again, it's only 2 microliters, and the sample or platform is completely 21 CFR Part 11 compliant. And we can see exactly how that can be useful as we manufacture our samples, and we can keep track of their quality. And that's the stunner in short. as you make your way through screening for stability and formulation, right, we're going to want to have the right tools again. So here I'm going to show you a few well-known cricket players, okay? And I'm going to use that as an analogy again to show you different instruments and how each of them bring unique tools or unique skill sets to the game, right? So the one here that I'm going to emphasize is going to be the Big Tuna. So let's take a step back. What we've done so far is we started with a large number of candidates, right? We've gone through a number of construct screening tests, and we're able to down select the right ones if you want, and we have a smaller number that we want to then take to develop and screen for the right formulation. And so if you have to imagine being able to screen through, let's just even say just 12 formulations, right? So if you have maybe 3 candidates, those are 36 different buffer exchanges that you have to do. That's both time consuming, right? And also not always easy to scale. And so if you could automate that, you could see the benefits of doing so pretty readily. And this is how the Big Tuna is going to be useful. So the Big Tuna is a very nice and quick platform to use. It's super easy, very low sample requirement in some cases. And the hands-on time is also quite low, as you can tell, just about 30 minutes. And you can get back to doing the actual work that can make a difference. We can do as many as 96 exchanges at once. The sample volume can be as small as 100 microliter, and we'll see why, up to 8 milliliters, so quite a dynamic range in terms of what can be handled. And while we can handle very high concentrations, the lowest concentrations that we've handled on the Big Tuna are as low as 0.05 mg per ml or 0.1 mg per ml are also typical on the low end. If you don't have a lot of highly concentrated samples, you can handle either one at either end of your concentration range. And so one of the unique things about it are the actual sample holders. So we call those plates unfiltered. And so they're essentially made of regenerated cellulose, and they are high pressure filtration plates. There are 2 types. The 96 well one can essentially accommodate 100 to 450 microliter. And you can see the different cutoffs that we have set there. So anywhere from 3 up to 100 kilodaltons, so different molecular weight filters there. On the right-hand side, you have the so-called unfiltered 24 or a 24 well plate. And this one can go from 450 microliter up to 8 mil in volume for each well. Right, so quite the range and lots of options for any person looking to buffer exchange in different levels of essentially throughput or capacity. And this is really low cost. This is in fact less expensive than Amicon filter per well or per sample. So it's really economical in that sense. The way we do buffer exchange is by doing one of 2 things. We can take your sample and reduce the volume and simply adding the new buffer that you wish to exchange into it. And as we do so, we'll repeat the cycle until we fully exchange into the new buffer, right, arriving to your final sample. And then you can concentrate at the end, completely optional to you. If you simply want to concentrate, so you've already done your buffer exchange, perhaps using a different method, and you want to find a quick way to concentrate, the Big Tuna can do that for you as well, independently of the buffer exchange. So you can do one or the other or both, right? It's really, really fast. And so if you're thinking about buffer exchanging your samples, those are examples that you can see here that all of them were done essentially in less than 1 hour of runtime for the most part. And you can see how quickly you can get through some of the samples. And as you increase the concentration, it may take a little bit longer, but relative to standard methods, this is still much faster than anything else that you can do. And remember that this is completely hands-off, right? So you walk up to the instrument, you put all your stocks essentially, and in less than 30 minutes, you click start, walk away, and the system will do all of the exchanges for you, and you'll pick up your results after. So let's walk through examples of this. This is a formulation screen that we did for one of our customers. And so the example here, right, is looking at the impact of pH and buffers, right? So we have one sample, one main protein of interest, and we're going to exchange this one into 12 different buffers of various pH values, as you'll see in the next table there. And so we've gone from pH 6.3 all the way down to a pH of 7.4. We decided to use a volume of about 4 mil at the beginning. And so you can see the average volume measured at the end. And so less than 0.01% difference relative to our target. The initial concentration for the samples were about 45.8 mg per mil. And you can see that at the end, we are very close to that at about 45.3 mg per mil. So again, well over 95% sample recovery is typical for those runs. You can see obviously in the example here that we are much higher than that. And the exchange rate here again is excellent. So you're going to have a very high efficiency when it comes to buffer exchanging your samples. So you can be assured that at the end, what you are getting is in fact a truly exchange or buffer exchange sample into a new buffer that you can then assess for aggregation and other indications of stability. So we then took those samples and we characterized them using the stunner. As a reminder, the stunner measures both concentration and DLS. So what you're viewing here in the left columns are essentially the samples in triplicates, right? And so I've simply highlighted the second one with a blue box to make it easier to read. You can see that we started about 45 mg per ml or so, and this is pretty consistent for our triplicates here. And then at pH 6.3, 6.8, and 7.4, we end up at pretty much the same concentrations throughout the exchanges there, okay? We look at the impact of the buffer exchange, or specifically look at the impact of the pH on the quality of your samples, right? So by the end, you're starting at 5.49 nanometers or so, and you can see that there are pretty significant differences in size based on the actual pH in which your samples are in fact stored. And so this is the beauty of this platform, but it's the beauty of using the two of them together, right? There's two players that have complementary skill sets, is that you can assess the concentration, and that's important, but you can also know exactly the quality of your samples and assess exactly how those things can be impacted by your conditions. So let's see exactly how the Big Tuna plays into that, right? So we talked about R&D, so early development for high throughput sample prep if you want to. And so if you have multiple samples to screen into one buffer, right, that's something that we can do pretty easily. Or formulation screening where you have a small number of candidates that you want to exchange into multiple buffers. As we've seen, up to 96 conditions can be screened per run or per experiment. Okay. So you've made your way down. You now have perhaps one major lead or one final sample that you have, and maybe a couple of backups, right? And so now you're ready to scale up. You're ready for the full production. And so again, this is the time where you really need to step it up and really kind of differentiate yourself from the competition if you want, and as you start getting into the end game, so to speak. So this is when the Hound, another platform, can be useful. So the Hound is an interesting platform that is really useful for morphology. So if you're looking at the shape and also getting an idea of the count and size of the particles in your sample, that platform can do so. And also we've added the ability to do chemical identification. So you can also find not only what kind of samples make up your drug, but also determine exactly where some impurities or contaminants may be coming from, right? So what I'm going to be showing you here is an example of an image taken by the Hound, and you can see that the actual sample itself or the platform can automate the image capture, but it can also give you exact information on the size of the samples, begin to group them into different size bins or size buckets if you want. This can be done automatically by the software, or they can be user-defined, completely up to you. You can pick your range of particles that you want to count. So you may say, only show me things that are visible, right? So above a certain size, from one range, one number to another, and they will only show you those. Or you can say, show me everything and put them into different buckets. And the sample and platform, the sample distribution, can be completely automated using the Hound, and we can see exactly how that can be useful. Finally, as you identify those particulates, you may then begin to attempt to identify them and determine exactly where they come from. The platform comes with a built-in library or a series of reference vectors that you can use in databases. You can certainly create your own or begin to further build up your library as you begin to characterize more samples. And so if you have samples that are unknown, you can use the database that is there. If you have samples that are unique to your production, you can store them and in the future easily identify them and figure out which components are present in your sample. And so let's take a look at this, right? So if you wanted to determine perhaps, what exactly is making up your protein aggregates, you can do that pretty easily. And if you have other contaminants as well, you can certainly identify them and have a chemical fingerprint for those. So I'm going to walk you through an example of this, right? So where a customer reported to us that they had a batch failure, meaning that they had produced a significant amount of their therapeutics. They went through the purification steps, and in the end, they found some impurities, right? And they could not figure out where exactly it's coming from. And so they found this cellulose fiber, right? And that caused 3 batches to fail. And when it comes to batch failures, right, we're really saying here, because we are so far downstream, we're looking at very high costs, essentially, and very expensive limitations when you have to stop your production and clean your bioreactor. And so they can identify the cellulose using the Raman spectroscopy that's built into the Hound. And so that's quite useful. But if you look at them carefully, you can see that there are some differences here, right? Where you have the raw one, you have the process results, and then you have the reference. You can easily identify where exactly it's coming from. But we do something that's a bit more sophisticated if you want. You can also use that to identify the root cause, because you can get cellulose from a number of different sources. But by being able to uniquely find the characteristics of each of those components, they're able to actually found that it came from the paper towels which were used to clean the inner wall or the inner surface of the bioreactors. And by simply changing the way they clean their bioreactors, they're able to eliminate some of those cellulose fibers, and therefore future batches were not compromised, and they were able to continue their operations without much interruption. Yeah. And so as you think about the Hound, right, I want you to encourage you to think about where exactly it makes sense. And so if you're thinking about how do I characterize my particles by size and composition, how do I determine the actual count of those samples as I make my way downstream, this is where the Hound can be useful. You can identify proteinaceous particles, right, not just synthetic ones, but you can also have the ability to store some of your reference spectra and use that to further assess stability and storage studies over time. And so it's really, really unique and very flexible in a sense, because you're getting both imaging, but you're getting intelligent image analysis combined with identification to be able to identify not only what's showing up, but where exactly it's coming from because of the Raman spectroscopy and the other lasers present. We offer you the ability to also look at visible particles, right? Not just subvisible, but also large particles that can impact the production process. And again, look at the aggregate composition. And finally, if something is flagged, you can actually make your way back and know exactly where it's coming from and remove it as early as you can. So if we now think about what we just went over, which is a pretty sophisticated antibody development process, right? So at every stage, things begin to be a bit more interesting where you have less samples, but the value of each strain is actually pretty significant. The further you make your way downstream, the more critical every step is, the more expensive or costly every limitation or stoppage or interruption can be. And so being able to have the right tools at any stage of development is critical, but more importantly, being able to carry the same tools from the beginning of your development all the way downstream can prove to be very advantageous, because you don't have to necessarily go back and learn new technologies. The same technology that you're using downstream can be communicated upstream, and you don't have to repeat the same experiments over and over again if something happens, because you are using the same tools all the way downstream. And so you can see that the uncle and lunatic are very great as you're beginning, but as you make your way downstream, the standard can prove to be more useful at different stages. And finally, we can add the Hound and the Big Tuna to improve some of the processes and the workflows that you may have commonly downstream. Okay. So I want to thank you for attending the talk today. I'm going to also refer some of you to your local distributors. So we are available to you at any stages. And again, if you have any questions, feel free to reach out to the members that you see on the slide here. As we do so, I want to also give you an indication of where you can find us. Our technologies are present among some of the top producers out there of biologics and biopharmaceuticals. And so you can rest assured that you are in good hands, and you're going to be members, if you want, of the Unchained group of users. And that's a pretty good group to be a part of. So thank you, and then we'll take any questions that you might have at this stage.
Thank you, Rifar. That was a great overview and how the Unchained tools can be used for really every stage during the antibody development. We've got some great questions here, and so I want to kind of go ahead and start asking you those questions.
Sure.
So one is for Big Tuna, is there going to be any protein loss when you do the buffer exchange, or do you know how much protein you might be losing in the process?
Not much at all. So in average, I think we tend to average over 95% recovery, right? So it's a very efficient buffer exchange process. And because our system is controlled, you know exactly where your protein is. It's either above the filter or it's above the filter. And so for the most part, you're not going to have to worry about protein or material loss as you would for other techniques. Okay.
So then also about Hound, so you can create a database, correct? Like how is that done? How long does it take? Is it done commonly?
Sure. Yeah, it's pretty routine if you want to. So the system does come with a very, very generous database. But if you want to add additional spectra, you can. So you can simply take some samples, characterize them, and store them and begin to build your own library. And that's going to depend, of course, on the number of samples that you want to characterize, but it's pretty quick and pretty easy to do. So it's a very simple process that's been optimized to make it easier for users.
Thank you. You had mentioned that systems fit perhaps in certain steps of antibody development, but has Unchained placed all these systems in these different steps and groups as well?
Yes, absolutely, yes. So without being too specific, I showed you examples of a number of companies which have our systems. And so those companies have our systems at all of those stages of development. So from very early stages, and you saw the example of Janssen, doing some of the construct screening, and other examples here of customers who are using them all the way down to manufacturing for quality controls, or even in regulated 21 CFR Part 11 compliant environments. Absolutely.
So right now I have just one question left. I will ask it, but in the meantime, if anybody else has something they want to ask, and please just put it into the Q&A box so we can still answer a question. Last question I have here, Wifar, is what is a good use for a stunner in a high throughput situation?
Sure. I think it's about protein quality control, right? And so when you think about high throughput screening, you're going to essentially go through a number of different conditions, different pHs, different excipients, right? You may look at different stresses, essentially sugars, so on and so forth. And so what you want to be able to do is assess the impact of those conditions on your sample's quality very quickly. The stunner can screen as many as 96 samples in one plate. We can actually run as many as 10 plates per experiment. So those are 960 samples or 960 conditions. If you wanted to, that you could screen both for concentration, but also for dynamic light scattering. And so that's really one of the most effective ways to use it. It's a very nice way to only use 2 microliter of sample, but gain as much information as you can about the quality of your samples as you buffer exchange them.
Okay, so thank you, Wifar. Thanks for answering all those questions so nicely. I think that is probably it for today. I want to thank you all very much for joining us. We will have a recording of this available on YouTube later. We will send you a link as well, and just in general get in touch with you. So if you would like to have a deeper conversation with us, you will know how to get a hold of us. And we would love to connect with you, and that can be us at Unchained directly, or as we had mentioned, with one of the many different distributors in your respective regions. So thank you again for attending this virtual seminar, and I do hope you have a great rest of your day.
Thank you.
Thanks.